Hello my dear friends, welcome to Live Prep. In the previous lecture, we studied about the individual structures of nitrogenous base, sugar and phosphate because these are the three components that we are studying under nucleotide. Nucleotide consists of these three components. What are those? Nitrogenous base, sugar and phosphate. Already within the nitrogenous base, two types, purines, pyrimidines we have studied. Within the purines again, adenine, guanine we have studied, and then under the pyrimidines we have studied about cytosine, uracil and thymine. Yes or no? And separately for the sugars we have studied that two types of sugars, ribose sugar, deoxyribose. In the deoxy also it is 2-deoxyribose. And separately we studied about the phosphate group, it is also called as inorganic phosphate. With structures everything we have studied, clear? And now what we need to do, we need to combine this. By combining these, we will get new other names, and first what we will do is, first we will see what are the names, understood or not? And please remember these names were given even in biomolecules of class 11th and now in this MBI of class 12th also. In both the cases they give. So we need to know all these things. And another important point is, in the NCERT sentences, they have given the sentences in such a way that how exactly these bondings are taking place between these two different molecules, and which molecule or which atom of this one is involving in bonding with this one. So each and everything was very clearly given. So that is the reason why we need to understand the structures very clearly. Please remember this. Okay, so that is the reason why, and now we are going with this particular RNA first. With respect to RNA, first we need to understand this particular table. Don't worry, I will explain each and everything. In the previous lecture, we studied about nitrogenous base, different types, and then we studied the structure of sugar separately. And when we are adding these two, that means if nitrogenous base is attached to sugar, it will form a molecule called nucleoside. Please remember it is SIDE side, it is not TIDE tide. It is forming nucleoside. Okay, and to this nucleoside, if phosphate group is attached, now it will form nucleotide. That's why nucleotide is having three components. What are those? Nitrogenous base, sugar and phosphate. Three components are present, that is nucleotide. If only nitrogenous base and sugar, only these two are present, that is called nucleoside. So what are these names, how we are writing all these names? Anyhow, this is the table conclusion, but now I will explain each and everything with more details. Okay, let us try to understand the names first. How actually the names were given? Let us observe this carefully. First we will write with respect to RNA. With respect to RNA. So first we need to talk about a nitrogenous base. Nitrogenous base. Yes or no? What are the various types of nitrogenous bases that are present in the RNA? What are those? First one, adenine, adenine and guanine. These two are purines, right? Adenine and guanine are purines that are present in RNA. Next one, with respect to pyrimidines, what are present here? Cytosine, cytosine. And the next one is very important because adenine, guanine, cytosine are present in both RNA and DNA. But what is the fourth one? With respect to RNA only we want. So it will be uracil only. Uracil is present only in RNA. It is not present in DNA. So that's why we need to write about uracil here. So this is the uracil. So this is what we have written for the nitrogenous base. And now we need to write about the sugar. What is the sugar that is present? Sugar that is present. Now let us write. In the case of RNA, what is the sugar present? Only ribose. So everywhere we need to write about ribose only. So here it is ribose. Ribose, the same ribose everywhere. Okay, simply I am keeping this ditto. So please observe this. Ribose sugar. And now what will form? Nitrogenous base plus sugar. Now what it will form? Nucleoside. Please observe this. Nucleosides are having two components. One is nitrogenous base and other one is sugar. So that it is forming nucleoside. Now we need to write the names. Please observe this. Adenine and guanine are purines. Purines. Cytosine and uracil are pyrimidines. Yes or no? Please remember this. Based on these purines and pyrimidines, namings are slightly changed. So that's observed carefully. Only nitrogenous base is adenine. If ribose sugar is attached to this, how the name will be changed? Please remember here, adenine. Up to this it is common, adenine. Adenine. And after this, so since it is nucleoside, you need to give a different name. If you are writing as it is adenine, you will be confused, right? Adenine is already nitrogenous base, and here you cannot write adenine. So the scientists have changed the name. What they did? They have added osine. O-S-I-N-E, osine at the end. Understood or not? So adenosine. So in this way, adenosine is nucleoside. Please observe this. Adenine is nitrogenous base, whereas adenosine is nucleoside. In this way, slightly the name will be changed. So if they are giving some list of names, based on the names you need to understand whether that is nitrogenous base, nucleoside or nucleotide. So that's why names are very important, and these names were very clearly given in NCERT. So it is part of NCERT, you should know this. Clear? Come back to the second one. Guanine plus ribose. This is also purine, right? For both the purines, osine will come at the end. Now can you guess what is the name here? Guanosine. Guanosine. It is the guan and osine at the end. Osine. Guanosine. So that's why adenosine here, guanosine here. Next one here. Here. Cytosine plus ribose. Can you write cytosine here? You cannot write osine here because already in the normal name itself osine is present for this. Please observe this one. Here cytosine is not nucleoside. It is nitrogenous base itself. Please observe this difference. Adenosine nucleoside, guanosine nucleoside, but cytosine is nitrogenous base itself. It is not nucleoside. So if ribose is attached, can you write cytosine again? No, because both the names are same. So that's why what scientists did is, for the pyrimidines, they have given a different ending here. Okay, different suffix here. What they did. So here it is cytidine, I-D-I-N-E, idine. Observe this. Cytidine. Understood or not? So in the same way, here for uracil also. Uracil plus ribose. Idine should be coming here. So here it will become uridine. Uridine. Idine is coming at the end. So in this way, with respect to purines, nucleosides will have osine at the end. That's why the names are adenosine and guanosine. With respect to pyrimidines, idine will come at the end. That's why it is cytidine and uridine. That's it. This is what about this particular content. So with this, nitrogenous bases plus sugar that are forming nucleosides are completed. And now what we need to do? So with this, this is over. Now to this nucleoside, now let us add Pi. Now let us add Pi one more. Pi is what? Inorganic phosphate or phosphate group. So phosphate group, you can also call it as a phosphoric acid. Understood or not? That means you are attaching an acid group to this one. Inorganic acid. Phosphoric acid is attached to this. Please observe this. Nucleoside is having two components. What are those? Nitrogenous base plus sugar. And that means nitrogenous base plus sugar, and now you are adding Pi. So that means three components will be there. So what it will become? It will become nucleotide. It will become nucleotide. Please observe this carefully. So now what we need to do? Adenosine plus Pi. What is the name? Guanosine plus Pi. What is the name? Cytidine plus Pi. What is the name? And uridine plus Pi. What is the name? So change it. So now we need to add this acid. So we need to write acid at the end. Yes or no? You are adding the acid. So this name will be slightly changed. And here what happens? For all these four, ileic acid will come at the end. Please observe this. So here it is adenosine is now, after attachment of Pi, it will become adenylic acid. Adenylic acid. Please observe this, ileic acid. For all these four, ileic acid will come. Simply you can write this. Guanosine plus Pi, what it will form? It will form guanylic acid. It will form guanylic acid. So here also ileic acid is coming. Next, cytidine plus Pi, here also ileic acid. Cytidylic acid. Cytidylic acid. Okay. Next one, uridine plus Pi, it will be forming uridylic acid. Uridylic acid. This is what. In this way, only nitrogenous base, sugar. Combinedly they are forming nucleoside. If Pi is also attached, now all the three components will be there, so that they are forming the nucleotides. So now corresponding to each one, you should know the name. Let us quickly go through this. Adenine is a nitrogenous base, plus a sugar it is forming adenosine. And to this, if Pi, phosphoric acid is attached, now it will form adenylic acid. So adenine is nitrogenous base, adenosine is nucleoside, Adenylate acid is nucleotide. These are the names. Same way with respect to guanine. Guanine is nitrogenous base. If ribose sugar is attached, it will form guanosine. And to this, if phosphoric acid is attached, it will form guanylic acid. So guanine is nitrogenous base, guanosine is nucleoside, guanylic acid is nucleotide. Next one, cytosine plus ribose, it is forming cytidine. To this cytidine, if Pi is attached, now it will form cytidylic acid. So nitrogenous base is cytosine and cytidine is nucleoside, cytidylic acid is nucleotide. Clear. Next one, uracil plus ribose is forming uridine. If Pi is attached to uridine, it will become uridylic acid. So uracil is nitrogenous base, uridine is nucleoside, uridylic acid is nucleotide. So in this way, these are the names that you need to understand. This is what the complete table, and all these names were clearly given in NCERT, in the NCERT. So the same thing I have written here. Is this clear? Observe it very carefully, the same thing we have written. Please remember, since we are talking with respect to RNA, ribose sugar we are talking. So that's why we should not talk about the thymine, because thymine is not present in RNA. Clear? In the same way, once you understood all these names, the same type of table we need to make for DNA also. But here there is a small difference. What is the difference? Adenine, guanine, cytosine present as it is. But instead of uracil, thymine is present. This is one difference that you need to notice. Second difference, with respect to sugar. In the case of sugar, what happens? In the case of RNA, it is simply ribose. But in the case of DNA, 2-deoxyribose sugar. Please observe this. When you are writing sugar individually, you need to write 2-deoxyribose. And now what about the nucleoside? You need to attach this. And once we are attaching, then what happens? Here the nucleosides will be formed. Okay, I will tell you the names and everything. And next one to this, PA is attached, so that nucleotides will be formed. And here what is the change? If you know the previous table, it is very easy to remember. So I will tell you the logic and everything, and we will go with that. Okay. And now first we will complete that explanation, and then we will come back to this particular table. Okay. This is the same table, even for DNA also. For the DNA, what we need to do? Instead of this R, we need to write D. Okay. So changes, let us focus here. What are the important changes that we need to focus here? Okay. Now let us see this. In the case of DNA also, nitrogenous base, adenine is present, guanine is present, cytosine is present. But what about the uracil? Uracil is not present. So remove the uracil. In place of uracil, we need to write thymine. In place of uracil, we need to write thymine. Is it clear? Nice. Next one. Come back to the sugar. What is the sugar that is present in the case of DNA? It is 2-deoxy. It is 2-deoxyribose. 2-deoxyribose, 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 2-deoxyribose. Please observe this carefully. You can write simply deoxyribose, or specifically you can write 2-deoxyribose, because deoxy is at second carbon position. Already in the previous lecture we studied this. Over. Next one. Nucleoside names. See here. Adenine plus ribose, adenosine. Fine, no problem. But it is not just ribose, it is 2-deoxyribose. So you need to make it as 2-deoxy. 2-deoxyadenosine. Please observe this carefully. So same way, everywhere you just add 2-deoxy. So here it will become 2-deoxyguanosine, and here it will become 2-deoxycytidine, and here instead of uridine, since it is thymine, here what will come? Here 2-deoxythymidine will come. Thymidine will come. Understood or not? Simply one by one we are studying, so please observe carefully. So this 2-deoxy we are adding everywhere. This original name is like normal only, so you can easily remember this. But here there is one technical detail, observe carefully. If you are writing the sugar individually, it will be 2-deoxyribose. But once you are attaching this, what is this nucleoside? Nucleoside means nitrogenous base plus sugar. Both of them are attached. That's why it is called a nucleoside. Once they are attached, now you cannot say it as a 2-deoxy, you need to say 2'-deoxy. Slowly I will tell you why you are keeping prime and all. So now what you need to do, it should be 2'-deoxy. Understood or not? Once again I am telling you, listen very carefully. If you are writing individual sugar, you can write 2-deoxyribose. But if you are talking about the same sugar position, same carbon in the same sugar, as part of nucleoside or nucleotide, in these what happens? Sugar is not present individually, that is attached to nitrogenous base. Since sugar is attached to nitrogenous base, to differentiate, I will tell you all these logics slowly. So here we are giving 2' instead of simply 2. So that's why in the nucleoside we are writing all 2'. Same logic here also. Even in the nucleotide also. See here adenosine, PI, adenylate acid, fine, but it is 2' deoxy. Add the same 2' deoxy here also. 2' deoxy adenylate acid, 2' deoxy, 2' deoxy guanylic acid, and 2' deoxy cytidalic acid. And here it is not uridylic acid, and here it should be 2' deoxy thymidalic acid. So it will be 2' deoxy thymidalic acid. That's it. So if you know the table with respect to RNA, you can easily write the table with respect to DNA. So now let us quickly go with the conclusion. Adenine plus 2-deoxyribose forms nucleoside called 2' deoxyadenosine. If PI is attached to this, it will form the nucleotide called 2' deoxyadenylic acid. That's it. We have covered all the names. Clear? Same way here. Guanine is a nitrogenous base plus 2-deoxyribose forms 2' deoxyguanosine. If PI is attached to this, now it will form the nucleotide called 2' deoxyguanylic acid. Next one. Cytosine is a nitrogenous base. If 2-deoxyribose is attached to this, it will form a nucleoside called 2' deoxycytidine. If phosphoric acid is attached to this, it will form the nucleotide called 2' deoxycytidalic acid. Clear? Last one. Here it is thymine. Okay, thymine. If it is thymine is a nitrogenous base. If it is attached to the 2-deoxyribose sugar, it will form 2' deoxythymidine, that is nucleoside. If PI is attached to this, it will form nucleotide called 2' deoxythymidalic acid. That's it. Very simple. So in this way, with respect to DNA, with respect to RNA, if you are observing first with respect to RNA, complete table, you can easily modify this with respect to DNA also. So main logical point, what is that? So with respect to sugar, you need to write 2-deoxyribose. But with respect to nucleosides and nucleotides, you need to write 2' deoxy. Why you are keeping prime? Slowly we will study under the structures. Is it clear? So this is what the complete table, and the same table is this one. This one is the same table. Understood or not? So this is how we are writing all these names. All these names were very clearly given in NCERT. Slowly, once we are completing the structures and all, then we will go with NCERT sentences. That time once again I will quickly tell you all these points. Clear? With this, the basic information regarding the names completed. Fine. Names, now you understood. But the major problem is with the structures you need to understand, because in the NCERT they are saying that so and so position of so and so is attached with so and making so and so bond. How is that possible? So for that we need the structures. Already we studied structure of nitrogenous base in the previous lecture. Structure of sugar also we have studied in the previous lecture, and the structure of phosphoric acid or phosphate group also we have studied in the previous lecture. So we know all the structures of all these 3 components. Simply we need to add this. So while adding, what we will do? Do you need to make the structures for all these 4 types? Not needed. With respect to purines, we will take one example. With respect to pyrimidines, we will take one example. That's it. Based on that, we will make it. So in this way, by taking two examples, yes, we can complete entire information. You can easily understand with respect to RNA, you can easily understand with respect to DNA. Everything you can easily understand. The only thing we need to do is, now we need to observe all these structures. So here I will draw the structures and I will mention the bonds. So you need to understand the bond locations. Okay, so and so bond is present between so and so. So each and everything with detail you need to understand. So please remember, this is more like a chemistry thing, but still very important for examination. So you should compulsorily understand all these things. Clear? And now we are going with the structures. So slowly I will draw this. So that is the reason why I am saying that please practice the structures first. If you know the structures, if you are able to draw the structure without copying from notebook, that is very great. So you can easily understand all these points. Clear? And how I am drawing is, first I will take a nitrogenous base here. First I will take a nitrogenous base here. I will draw the structure of nitrogenous base here. And then here I will draw the structure of sugar. I will draw the structure of nitrogenous base here, sugar here. So once I am making a bond here, what happens? Nitrogenous base plus sugar will be formed. What is this nitrogenous base plus sugar? Nucleoside. According to that particular molecule, you will know the particular name. Already just now we studied the names, right? You can apply the name. With that, nucleoside is completed. And for the same nucleotide, what we will do? We will add one phosphate here, another phosphate here, another phosphate here. Understood or not? So that what happens? So nitrogenous base plus sugar is forming nucleoside. To this, if phosphate is attached, now nucleotides will be formed. So in this way, in the single diagram, we can understand all these things. Is this clear? Yes. And like this, two diagrams we will take, because here for the nitrogenous base, I will take example of one purine. And in the next slide, I will take the example of a pyrimidine. Simple logic. Clear? And what I will do is, I will try, first I will complete all this information. So based on this, we can go for the pyrimidine. First I will take the purine and I will complete this, then we can go with pyrimidine. Clear? Let us start with this. Simple example let us take. For the nitrogenous base, let us take the adenine. This is one simple example for us, adenine. Already we know the structure of adenine. Quickly draw this one, okay? So in the previous lecture, we studied about all these structures. So this is the structure of adenine, okay? Adenine is a 6-amino purine. So this is the structure of adenine, yes or no? So for your understanding purpose, I will give the numbers also. This is the number 1, number 2, number 3, number 4, number 5, 6, 7, 8, and this is the number 9. Yes or no? This is the structure of nitrogenous base. And then we need to draw the structure of sugar. Instead of drawing here, here I will draw it because I need to combine this. So that's why I will draw it here. So this is the sugar. So already I told you, whenever we are drawing the structure of sugar, we need to use furan ring. Yes or no? So here let us write this one. Okay. So the furan ring, let us draw it here. Understood or not? For this furan ring, and here we are talking with respect to, first let us take with respect to RNA. With respect to RNA nucleotides, it is, we are talking about only single nucleotide, not complete RNA. Please remember. RNA nucleotide means what type of sugar is needed? Ribose sugar is needed. And already we know the structure of ribose sugar. What we are saying? Here it is OH, and here it is H, and here it is OH, and here it is H, here it is OH, and here it is H, and here it is H, and here it is CH2OH. It is CH2OH. This is the structure of ribose. This is the structure of ribose. Here I will write ribose. This is adenine and this is the ribose. And for the ribose also, already we have studied the numbering. Yes or no? So this is the carbon number 1, carbon number 2, carbon number 3, carbon number 4, and this is the carbon number 5. Yes or no? So this is what about the adenine and the ribose. Nice. Individual structures we know. And now what is our aim? We need to combine this. Once you are combining these two, what happens? Nitrogenous base plus sugar, what is formed? Nucleoside is formed. Nucleoside is formed. If this is adenine plus ribose. Adenine plus ribose is adenosine. Suppose this is guanine plus ribose, guanosine. Understood or not? Suppose this is adenine, but this is 2-deoxyribose. 2-deoxyribose means what happens? Instead of OH, what will be there? H will be there. That is the difference. Suppose this is adenine, this is 2-deoxyribose. After attachment, what happens? 2'-deoxy will come. Yes or no? That's what happens. 2'-deoxyadenosine. Suppose this is guanine and 2-deoxyribose. 2'-deoxyguanosine. That's it. If you know structure related to one, and automatically you can understand the structure related to other one. Clear? And now let us focus on this particular content carefully. Observe very carefully. And here what happens? And here how the bond will be formed here. This is the purine and this is the sugar. And in the purine at the ninth position, what is the atom that is present? Nitrogen is present. But what is the valency of nitrogen? 3. But here how many bonds were mentioned here? 1 and 2. Only 2 bonds were mentioned here. That means automatically the third bond will be with hydrogen. Already we studied this point also. Automatically the third bond will be with hydrogen. And this is what about normal adenine? This is the normal ribose. And now where the bond will be formed? Hydrogen of N9 of nitrogenous base is is removed along with OH group of first carbon of sugar, maybe ribose sugar, 2-deoxyribose, whatever it is. Observe this carefully. Hydrogen of N9 of nitrogenous base, that is purine, and OH group of first carbon of sugar. Now these are removed as water molecule. H2O is removed. Water molecule is removed. Please observe this content carefully. Water molecule is removed. Now what happens? Now there will be a bond that is formed. Understood or not? Now the bond will be formed here. Now this as it is, I will draw in the next slide. So please observe carefully. Please observe carefully here. Directly I am drawing the adenine here. So this is adenine. So this is adenine 6 amino, right? So these are the positions of double bonds. And here we are drawing the ribose sugar. Ribose sugar. Since it is ribose sugar, at the second carbon position I am drawing the OH group. Okay. And here it will be CH2OH. Please remember, here I am not drawing hydrogens, still it is correct. And here what happened? Now directly it is connected. Yes or no? Observe this carefully. Here hydrogen, here OH, removed in the form of water molecule. Now what happens? Bond will be formed between N9 of purine and first carbon of sugar. Understood or not? The same thing we have made, N9 and first carbon of sugar. So now let us see this particular content. This is N9 and this is the first carbon of sugar. Understood? And now let us see this content carefully. This is the number 2, number 3, number 4 and number 5 carbons here. Yes or no? Now observe carefully. In this one particularly what happens? 1, 2, 3, 4, 5, 6. This is 7, 8 and this is the 9. We know the numbering. And now why you are keeping the primes here? Prime, P-R-I-M-E, prime. P-R-I-M-E, prime. So originally how you will mention? Like this we are mentioning. Understood or not? So this is the prime. Okay. So whenever we are saying that this is the 5 prime, 3 prime. So why we are keeping the primes? Now we need to clearly understand the significance of prime. So please listen very carefully. Why are you keeping the prime? If you are writing individually sugar, you are not keeping the primes. But once you are connecting these two, then you need to compulsorily keep the primes. Why? Because observe carefully. This is the carbon that is present in the nitrogenous base. Yes or no? This is the carbon present in nitrogenous base, carbon present in nitrogenous base, carbon present in nitrogenous base, and this is also carbon present in nitrogenous base. Yes or no? Now observe carefully. Suppose in this particular individual structures, suppose I am saying second carbon of nitrogenous base means this is the second carbon of nitrogenous base. Clear? In this second carbon of sugar means this is the second carbon of sugar. Clear? Since individual molecules, we are using the word called nitrogenous base and ribose sugar. Likewise, individually you are talking. But once you are connecting these two, will you say individually nitrogenous base, individually sugar? No. We are giving another name. Nucleosides are having the name, right? Adenine plus ribose, adenosine. Understood or not? Adenosine. Now the same question. Second carbon of adenosine. Which one you will say? Here also there is a second carbon and here also there is a second carbon. Now which second carbon you will take? Now I hope you understood what is the problem. So once you are attaching, the problem is second carbon, second position carbons, and fourth position carbons, fifth one carbon. So at some locations numbers are matching and they will cause confusion. Please remember, they will cause confusion. So that's why once these two are attached, now you need to change the numbering. Numbers will remain like that only, but some extra symbol we are adding so that we can differentiate them. And now for whom we are adding these primes? This prime we are adding for whom? For the numbers of nitrogenous base or for the numbers given in sugar? It is for the sugar. So that's why these numbers will remain like that only. So even though these two are connected, the numbering of nitrogenous base will remain same. But in the sugar what happens? This will be called as 1 prime carbon, this will be 2 prime carbon, this will be 3 prime carbon, this will be 4 prime carbon and this will be 5 prime carbon. So in this way primes are given only for carbons present in the sugar. Understood or not? So that is the reason why actually when we are talking about this particular table, see here, individually sugar it is 2-deoxyribose, but once it is attached, now if you want to talk about sugar of, sorry, second carbon of sugar means 2' deoxy, it is not 2-deoxy. Do you have to understand this? So what happens if you say 2-deoxy? After attachment, see, anyhow this is the ribose sugar, so no problem. Suppose here H is present, it is deoxyribose. Yes or no? So in that case, second carbon 2-deoxy means deoxy will be here. That is wrong concept. So we need to talk about deoxy at this position. At this position means what is this carbon? Is it second carbon or 2' carbon? That is 2' carbon. This is the logic. So please try to understand the concept with logic so that you can easily understand the NCERT sentences. So this is what the logic is. So what is the overall conclusion? If we are talking individually, individual numbering we are giving. But once they are attached, what happens? The numbering of nitrogenous base will remain same. But for the carbon numbers of sugar, we are keeping primes to avoid confusion. Is this clear? That's it. In this way, what happens? This is the adenine. This is the adenine. And this is the ribose. This is the ribose. Understood or not? Adenine plus ribose. Now tell me what is the name of this? What is the name of this entire structure? Adenine plus ribose, entire structure. What is the name of this? Adenine plus ribose. Just now we have studied. What is the name of this? Adenosine. Now this is adenosine. This is adenosine. I hope now you understood how to write these names and how to write the structures also. That's it. Very simple. Clear? Next one. Now with this adenosine is completed. Now to this adenosine, if you are adding one phosphate group, what is that phosphate group? H3PO4. You can write H3PO4 or simply you can write H2PO4 minus, anything you can write. So now let us write P double bond OH, OH, and here you can write OH or you can write O minus, anything you can write, so no problem at all. Okay. So in this way let us write. Okay. So H3PO4, with respect to H3PO4 I am writing. So this is the phosphate. So adenine plus ribose combinedly formed adenosine. To this adenosine, if phosphate group is attached, then what happens? Observe this carefully. Now here what happens? Now we need to add here, right? So here let us add this. So adenine ribose sugar, sorry, nitrogenous base adenine and ribose sugar completed. And here we need to write this phosphate group. P double bond OH, OH and OH. Now what happens? Now this phosphate group should be attached to this. And how exactly it will attach? Observe this carefully. Okay. OH, many students are making mistake here. In the biology, this question is not important. But in the chemistry, it is very important. Now here also water molecule will be removed. Water molecule will be removed. Please observe this very carefully. What is correct concept? This is the correct concept. OH of phosphoric acid and H of this alcohol. CH2OH. OH means what? Alkali group, right? So this hydroxyl group. From that H and from the phosphoric acid OH will be removed in the form of water molecule. Here also H2O is removed. H2O is removed. And what many students are thinking, so what is the mistake done by students? They are removing OH from this and they are removing H from this. This is mistake. Please observe carefully. So OH from phosphoric acid and H from this alkali group will be lost in the form of water molecule. So that bond will be formed here. So that point we need to connect here. So how you are connecting this? So you need to remove OH from this and you need to remove H from this. So that now you can connect these two. This P will be directly connected to this O. Understood or not? So now what is the name of this? Now what is the name of this? Nitrogenous base plus a sugar, that is called nucleoside, plus phosphate, that means up to this. What is the name of this structure up to this? It is called as nucleotide. What is that nucleotide name exactly? Adenine plus ribose is adenosine. Adenosine plus phosphate, it is called as adenylic acid. You can write this as adenylic acid. Adenylic acid. That's it. Simple. So in this way, nitrogenous base, sugar and the nucleotides names will be derived. Very simple concept. Very simple concept. If you know the structures clearly, you can understand each and every point very clearly. Is this clear? Now, suppose this is the guanine. Guanine plus ribose, guanosine. Guanosine plus phosphate, guanylic acid. Completed. Understood or not? Now with respect, because let us study purine separately. Suppose this is adenine only. Suppose this is 2-deoxyribose. 2-deoxyribose means what happens? Here instead of OH, H will be there, that's it. So now what happens? Adenine plus 2-deoxyribose. Now combined it is called as 2'-deoxyadenosine plus phosphate, 2'-deoxyadenylic acid. Here we are writing with respect to RNA. Once you know for RNA, you can easily write for DNA also. That's it. Very simple structures. With this adenylic acid is completed. But generally, we are not stopping with single phosphate. Now let us come back to this. We are not stopping with single phosphate. In some nucleotides, what happens? Nucleotide, to say that it is nucleotide, at least one nitrogenous base, one sugar, one phosphate should be there. But some cases, what? This is a nucleotide. But in some nucleotides, what happens? Two phosphates are present. Nitrogenous base plus sugar plus two phosphates are there. In some case of nucleotides, what happens? Nitrogenous base plus a sugar plus three phosphates also present. So that's why we will add these two also here itself, so that we can write the name. So now what we need to do here, we need to write one more phosphate. So write one more phosphate here. So now what happens? Now once again water molecule will be removed like this. Once water molecule is removed like this, how you need to change this here? Now you just add that one. Now P double bond O, OH, OH and OH. So now what we need to do? We need to remove the water molecule. So remove the water molecule so that now this P is directly connected to O. Understood or not? So this is one type of molecule. In another type of molecule, what happens? In another type of molecule, another phosphate is added. What is that another phosphate? Here you just add it. P double bond O, OH, OH and OH. Here also water molecule should be removed. Remove the water molecule and attach that phosphate group. And how it will become? Now observe this very carefully. Now here what happens is, here P double bond O, OH, OH and OH. And from this what we need to do? We need to remove this water molecule. Remove the water molecule. Once you are removing the water molecule, now what happens? Now this bond will be formed. Clear? Understood or not? So in this way, nucleotides means nitrogenous base plus sugar plus phosphate. In that phosphate also, some nucleotides are having single phosphate, some are having two phosphates, some are having three phosphates. Now how you will give the naming here? How you will give the names to this? So single phosphate, so monophosphate. Two phosphates, diphosphate. Three phosphate, that is called a triphosphate. So now for the same structure we will write the names. How you are writing the names? Observe this very carefully. Now up to here it is the nucleoside. What is that nucleoside? Adenine plus ribose is adenosine. Clear? Adenosine plus phosphate is adenylic acid. Clear? No problem. But here what we are writing, for the same adenylic acid we are writing a different name. Adenylic acid means single phosphate is present. So how you can write this? It is the adenosine having one phosphate. Can you write like this? Forget about this part. Up to this only you just see this. Adenine plus ribose, adenosine having single phosphate. So how you will write this? Adenosine single means what? Monophosphate. Adenosine monophosphate. In this way also you can write the name. So please observe this very clearly. Up to this part, what is the name? Adenosine monophosphate, single phosphate. But what is the mistake done by students? Many students are making mistake. The mistake is they are not calling it as adenosine monophosphate. Then what they are doing? They are saying that adenine monophosphate. If you are saying adenine monophosphate, you are not talking about sugar. That means you are making mistake here. So please observe the technical words. Sometimes they may, in the words itself they will give. You need to observe all these things with respect to spelling itself. You know the concept, but if you are not good at observing the spelling, unnecessarily you will face the problem. So please observe the spelling also. What is the correct name for this particular one? Adenosine monophosphate is the correct. If you are saying that adenine monophosphate, that is wrong. Understood or not? Because this phosphate is attached to ribose sugar of this particular adenosine or nucleoside. phosphate is not attached directly to nitrogenous base. So please observe this. So that is the reason why it should be called as adenosine. First you need to say nucleoside name of the nucleoside. Then you need to say how many phosphates. If one phosphate, monophosphate. I hope you understood this. And now let us see up to this. What is the name up to this? It is called as. Up to this means what? Nitrogenous base plus sugar means nucleoside. How many phosphates you are talking? 2 phosphates. So nucleoside plus 2 phosphates. So how you will write this? Nucleoside diphosphate. Understood or not? So what is this nucleoside? Adenine plus ribose is adenosine. This is the nucleoside. Adenosine. Adenosine is the nucleoside. Having 2 phosphates. Having 2 phosphate means diphosphate. Adenosine diphosphate. That's it. Over. Namings are very simple. Very simple. Only thing is you need to understand the logic behind the naming. That's it. Over. Suppose you are starting from that end to this end. That means here, from here to here. What is the name? Adenine plus ribose. That is the nucleoside. Adenosine. 1, 2, 3. How many phosphates? 3 phosphates. Then what will be the name from that end to this end? It will be adenosine triphosphate. Adenosine triphosphate. Adenosine triphosphate. That's it. Understood or not? And now we will see what are the standard abbreviations. What are the standard abbreviations? AMP. So it is called AMP. What is AMP? Adenosine monophosphate. And this is called ADP. Okay, ADP. Adenosine diphosphate. Understood or not? And this is ATP. Adenosine triphosphate. Already you studied, no? ATP is the energy currency of the cell. This is the ATP. This is the structure of ATP. That's it. Very simple. So each and every point with logic you need to understand here. That's it. Nitrogenous base plus sugar is forming nucleoside. Nucleoside plus 1 phosphate is you can say that so and so nucleic acid's name you can say, or else you can also call this as nucleoside monophosphate. If 2 phosphates are there, nucleoside diphosphate. On the nucleoside, if 3 phosphates are there, nucleoside triphosphate. That's it. Very simple. Understood or not? If this is related to adenine, adenosine monophosphate, adenosine diphosphate, adenosine triphosphate. That's it. So how we are writing these names? Adenosine monophosphate, adenosine diphosphate, adenosine triphosphate. Over. Suppose instead of adenine, if guanine is there, what is the change for guanine? In the guanine at second position NH2 and here double bond O is there. That is the difference. Remaining entire structure will be same. Then what is the name? Guanine plus ribose will be guanosine. Then guanosine monophosphate, guanosine diphosphate, guanosine triphosphate. Completed. So how you will write the names for this one? Guanosine monophosphate, guanosine diphosphate, guanosine triphosphate. In all these cases, what is present? Ribose sugar is present. Ribose sugar is present. Yes or no? Simply, let us get all the names from the single structure. If ribose sugar is there, these are the names. Suppose instead of OH, if H is present, then what it will be? Adenine plus 2-deoxyribose. Adenine plus 2-deoxyribose is called as deoxyadenosine. Yes or no? And now here it will be deoxyadenilic acid or simply deoxyadenosine monophosphate. And up to this it is called deoxyadenosine diphosphate. Up to this it is called deoxyadenosine triphosphate. Simply you need to add a deoxy. Understood or not? So with respect to deoxyribose, if deoxyribose is present, how the name will be changed? Simply add a small d. What is this small d? Deoxy. Deoxy adenosine monophosphate. Deoxy adenosine diphosphate. Deoxy adenosine triphosphate. Same way here also. If it is instead of adenine, if guanine is there, deoxy guanosine monophosphate. Deoxy guanosine diphosphate. Deoxy guanosine triphosphate. That's it. If you properly know for one structure, you can easily read for all others. Understood or not? This is what. So these are related to RNA because ribose sugar is there, and all these are related to DNA. At the same time, we are studying for both DNA related, RNA related, both the components. So these are the nucleotides. All these are nucleotides. whether single phosphate, 2 phosphate, 3 phosphate, whatever it is, these are nucleotides. That's it. This is how we are drawing the structures for these nucleotides. But here we used this nitrogenous bases, in that we used purines only. And now we will take one example of pyrimidine, and this time what I will do, directly I will make the bonds. Clear? Now let us quickly go through this. So here let us take a pyrimidine. Previously we took a purine, right? This time let us take a pyrimidine. So pyrimidine means let us take the cytosine. That will be easy because cytosine is present in both DNA and RNA. Yes or no? So in the case of cytosine, what happens? Here NH2 is there. This is the fourth position, and here double bond O is present. Yes or no? Cytosine is 2-oxy 4-amino pyrimidine, or you can write 4-amino 2-oxy pyrimidine. This is the cytosine. This is the cytosine. And below this, let us add the ribose sugar. First, let us talk with respect to ribose sugar. In the ribose sugar, again, here same bond. And what is the position of this one? This is the N1, right? Numbering also you need to know. Already we studied numbering. This is number 1, number 2, number 3. That is number 4, number 5, number 6. So in the case of purine, what happened? N9 attached to first carbon of sugar. But here N1 is attached to first carbon of sugar. Again, here also water molecule will be lost. And here since ribose sugar, OH present. And here also CH2, OH is present. And up to this what happens? This is the ribose sugar, right? This is the ribose sugar. Here I will write. This is the ribose sugar. Observe the concept. Same concept. Logics, everything are same. This is the ribose sugar. Now what happens? And here what happens? These two got attached. So cytosine plus ribose. What is the name? Cytidine. Yes or no? Cytosine plus ribose is cytidine. Completed. Now come back to the next one. So with this what happened? Nucleoside is formed. Cytosine plus ribose, cytidine. This is the nucleoside that is formed. Clear? Now phosphate group. What is the phosphate group? P double bond O, OH and OH. Now what we need to do? Remove the water molecule, OH from this, H from this. Remove it. And then you just make a bond here. Yes or no? So this is the bond between two molecules. Yes or no? Like this. So now what is the name of this one? Now its name is cytidine with 1 phosphate. Yes or no? You can call it a cytidine monophosphate. But what is the general name? Cytidine plus PA is called cytidilic acid. It is called cytidilic acid. You can also call this as cytidine monophosphate. So how you will write this? CMP. Cytidine monophosphate, CMP. Clear? If you are attaching one more phosphate, same way, one more phosphate. Like this, you just add one more phosphate. And here also you need to remove water molecule. So OH from this, H from this you just remove. And then you just make a bond here directly. And now what will be the new name for this one? Nucleoside with 2 phosphates, right? So now you just write the name here. So what is the name we are writing? Cytidine with 2 phosphates. So it is cytidine diphosphate. It is simply called as a CDP, cytidine diphosphate. If you are attaching one more phosphate, P double bond O, OH and OH. And here also let us remove water molecule. So OH from this, H from this let us remove, so that we can connect these two. So now what is the name of this particular molecule? Cytidine with 3 phosphates. So up to this entire structure is called as cytidine triphosphate. So simply it is called CTP. Observe each and everything very carefully. That's it. This is the cytidine triphosphate. Instead of cytosine, if uracil is there, uracil plus ribose, uridine monophosphate, uridine diphosphate, uridine triphosphate. Completed. Suppose it is with respect to DNA. Here instead of OH, what should be present? H should be present. Cytosine plus 2-deoxyribose, it will form 2'-deoxycytidine plus 1 phosphate. It is called 2'-deoxycytidilic acid or 2'-deoxycytidine monophosphate. Up to here 2'-deoxycytidine diphosphate. Up to here 2'-deoxycytidine triphosphate. Simple name. Naming everything are very simple, but only point you need to focus is location, bond location. Here N1 is involving in bonding. But in the case of purine, N9 is involving in the N9 is involving in the bonding with first carbon of the sugar. This is how the structures are possible. And here also same way let us write. Here what is the structure that is formed? CMP, CDP, CTP. Same names we will write here. Cytidine monophosphate, cytidine diphosphate, cytidine triphosphate. Instead of cytosine, if uracil is present, uridine monophosphate, uridine diphosphate and uridine triphosphate. And these are with respect to ribose sugar containing. If deoxyribose is present, small d will come. Small d CMP, deoxycytidine monophosphate, deoxycytidine diphosphate, deoxycytidine triphosphate with respect to deoxyribose sugar. And here with respect to thymine, please remember if it is deoxyribose sugar, uracil cannot be present, it should be thymine that is present. Then what happens? Deoxythymidine monophosphate, deoxythymidine diphosphate, deoxythymidine triphosphate. Simple names. And these are with respect to deoxyribose. That means these nucleotides are related to DNA and these nucleotides are related to RNA. That's it. Very simple concept. So what you need to understand, what are the various names that we are using? Names that we are using, and then what are the structures and where they are linked, what are the names, everything you need to understand. Clear? With this, these basic structures are completed, and there is a last content that is called as names of the bonds. Already we have given the entire bonds and all, but simply we need to add the names, that's it. I hope now you are perfect with this structures, right? So once you know about these structures, and now let us add the names. Observe this content very carefully. This is the nitrogenous base and this is the sugar. Between this is the bond, right? This is the bond. This is the bond that we are talking here. And what is the name of that particular bond? So since this is the sugar, right? Ribose is a sugar. Since sugar is involving in the bonding, it is called as glycosidic bond. Already in the biomolecules chapter, very clearly we have studied this. If sugars are involved in the bonding, that bond is called as glycosidic bond. Here also sugar is involved in the bonding, that's why it is called as glycosidic bond. Very simple. But what type of glycosidic? Glycosidic bonds are two types, N-glycosidic, O-glycosidic. Observe this. This is the sugar that is attached to which atom of nitrogenous base? Is it attached to N or is it attached to O? What type of molecule it is? What type of atom it is? So it is N-glycosidic. Mainly in the case of biomolecules, carbohydrate structures, there you are studying carbon-carbon linkages. They are also glycosidic bonds, but they are carbon-carbon linkages. But here this carbon is attached to nitrogen. Since it is attached to nitrogen of nitrogenous base, it is called as N-glycosidic bond. Please observe this, very clearly given in NCERT. The name of bond that is formed between nitrogenous base and sugar is called as N-glycosidic bond. This is the first point you need to observe, and exactly the same content applicable to this one also. Nitrogenous base and sugar. This is pyrimidine, right? Here also. But please observe, it is not N9, it is N1. That is the difference. But here also sugar is attached to nitrogen. So this is also called N-glycosidic bond. N-glycosidic bond. So in both the case it is N-glycosidic. Sometimes what happens is they will ask the question with more clarity. So within brackets I am writing. In NCERT they have not given. So we are writing with clarity. What is this one? N1. N1 is connected with, what is this one? Carbon first prime, right? So in this, what is the bond? N1 is connected with carbon C, carbon 1 prime. Like this also you can write, no problem. Simply you can say N-glycosidic bond, no problem, 100% correct. With more clarity if you are writing, in the pyrimidine N1 involved, so N1 with C1 prime, carbon of first prime. So this is the bond position. In this way also you can write, still it is correct. Suppose in the same way if you are writing for a purine, in the case of purine what is involved? N9 is involved. So what you will write here? N9 with, what is this one? C, carbon 1 prime. In the case of pyrimidine, N1. In the case of purine, it is N9. But whatever it is with N only. So that's why here also N-glycosidic bond, there also N-glycosidic bond. So in this way, between nitrogenous base and sugar, glycosidic bond is present. Clear? Next one. Next one, let us see. Between sugar and first phosphate. Between sugar and first phosphate. This is the organic acid, right? Whatever it is acid, and this is the alcohol. Between acid and alcohol, what is the bond that is formed? That is called ester bond. If it is organic acid, simply we are calling it as ester. But this is what? Phosphoric acid. Phosphoric acid and alcohol. What is the bond it is called as? This bond is called as phosphoester bond. It is called as phosphoester bond. Many students are making mistake. They are writing this as phosphodiester. That is wrong. It is not phosphodiester. It is phosphoester. It is single ester bond only. It is phosphoester bond. Fine. Next one. This is acid. This is acid. Between two acids, it is N-hydride bond. But what type of acids? Phosphoric acids, inorganic acids. Phosphoric acid, phosphoric acid. That's why it is called as phospho-N-hydride bond. It is called as phospho-N-hydride bond. Phospho-N-hydride bond. Locations, everything you need to clearly understand, because in the next lecture again the same content will be continued. Clear? Next one. This is also same, phosphoric acid, phosphoric acid. So same bond. It is phospho-N-hydride bond. It is phospho-N-hydride bond. This is what the names of the bonds. All these names were very clearly given in NCERT. So you need to remember bonds also. And what they said, nitrogen of this nitrogenous base is attached to C1 prime of sugar. Very clearly in sentence they give. So you need to understand. And the bond formed is N-glycosidic bond. And phosphate is attached to C5 prime. 5 prime carbon of means OH of 5 prime carbon of the sugar they gave. So you need to understand all these sentences. So to understand those sentences, you need to know about the position of all these bonds. So what is the overall conclusion? Between nitrogenous base and sugar, N-glycosidic bond. Between sugar and first phosphate, phosphoester bond. Between two phosphates, it is phospho-N-hydride bond. Here also it is phospho-N-hydride bond. Same names as it is we are writing even for pyrimidine also. Between nitrogenous base and sugar, N-glycosidic bond. Between sugar and first phosphate, once again it is phosphoester bond. Phosphoester bond, like previous only. Phosphoester bond. Next one, between two phosphoric acids, it is phospho-N-hydride bond. It is a phospho-N-hydride bond. Phospho-N-hydride. Here also between two acids, it is once again phospho-N-hydride bond. Phospho-N-hydride bond. Understood or not? This is how we are writing the names of all these molecules. So with this particular content is completed. So now here we will write one generalized information. Observe this generalized information. With respect to RNA, let us see means ribose sugar containing. What are the names we are writing? AMP, next GMP, CMP, and UMP. These are the various names we are using for monophosphates. And in the same way, ADP, GDP, CDP, and UDP. Yes or no? Next one, triphosphates. ATP, GTP, CTP, and UTP. These are with respect to ribose sugar. Same way here, D, AMP, D, GMP, and D, CMP, and D, TMP. These are with respect to deoxyribose sugar. Okay, these are related to DNA related, and they are having deoxyribose sugar. Same way here also, D, ADP, D, GDP, D, CDP, and D, TDP. And last one, D, ATP, D, GTP, D, CTP, and D, TTP. Overall, these are the structures that you will get. For all these structures, with the help of only those two structures, we have completed entire information. So if you want to practice it, because you need to understand all these with structures, and what are the carbon positions, what are the molecules that are removed, what is the name of the bond that is formed. These are the things you need to remember. So these are the things everyone should know. Every student should know about all these things with structures. Then only your concept of this particular lecture will be perfect. I know this is more structure related. Many of you are getting confused. Try to watch the video once again if you are having any problem, but please practice all these things. You need to know the structures. If you know individual structures, entire content will be very easy. It will be on your fingertips. So please practice it very well so that you can get the content. Now let us come to the conclusion. How you will write this one in general? In general, what you are writing? How you will say nucleoside monophosphate? All these names, if you see, it is not nitrogenous base monophosphate. It is nucleoside plus monophosphate. So NMP, nucleoside monophosphate. This is the generalized word. This is the nucleoside diphosphate, nucleoside triphosphate. In this way we are writing. Here what you will write? You will add small d, deoxy nucleoside monophosphate, and here deoxy nucleoside diphosphate, and this is deoxy nucleoside triphosphate. In this way, in general we can call, instead of calling all these separately, simply you can call like this. So these are the names that we are using, and slowly in the next coming lectures we will use these particular things also. That's why we are studying about all these things here itself. Clear? And better we will stop it here. Anyhow, in the next lecture what I will do is I will revise these bonds locations once again so that we can go further with the numerical calculation. Please observe this. Why I am talking about all these bonds here? Because based on this, in the next lecture we need to go for bonds and calculations. So that is very important and little bit confusing once again. So please practice the structure so that this lecture will become very easy. You need to remember these names with structures and bonds. So please practice this. It will take time, I know. So please take a little bit more time and practice everything and be perfect by the time we are going for the next lecture. Clear? So with this we have completed this particular one. Please remember, in the DNA uracil is not present. Clear? We have completed the structures of nucleosides, tides and everything with the names and everything. Clear? With this particular lecture is completed. In the next lecture we will talk more about the bonds calculation and all the things. Okay. Thank you so much, my dear friends. We will meet in the next lecture. Thank you.